So in microevolution, what changes from one generation to the next? It's the frequency of something. I'll just put on the chat. Alleles. Yeah, the frequency of alleles. So macroevolution is an accumulation of change over generations and generations. And it also, it also requires something called new ecological opportunities. So remember that term, new ecological opportunities. Speciation, it's an accumulation of changes through mutation, genetic drift, sexual reproduction, all those things change allele frequencies from generation to generation. Natural selection, genetic drift, gene flow, mutation. But we're just talking generation to generation. That, you know, we're not a new species compared to our parents, although sometimes they might think so. Depends how well they understand you, I guess. But yeah, it happens over long periods of time. Yeah, the mystery of mysteries. Darwin explored the Galápagos Islands, as we know. He discovered plants and animals that are found nowhere else on Earth. And of course, his first question would be, just like your question would be, how did they get there? How did they get there? This, for example, this is a flightless cormorant, a Galapagos cormorant. And you look at the Galapagos cormorant and you go, oh my gosh, look at those wings. He must be damaged in some way, but it's not damaged in any way. The cormorant just has reduced wings. It doesn't fly anymore, just like penguins. Penguins don't fly anymore either. Are the wings of any use to the animal? Sure, sure. It swims with them. Cormorants are amazing underwater swimmers. We have two species of cormorants here now, the pelagic cormorant and the double-breasted cormorant or something like that. And anyway, they're you can see them off the water. One species is off the water, one species is often in the Lost Lagoon. And you'll see them drying, like this one is drying its wings. There's one thing about cormorants, which is different from other water birds or waterfowl, is that it doesn't have oil. It doesn't have oil in its feathers or in its skin to extrude onto its feathers to prevent getting waterlogged, essentially. So it has to dry its wings that way. Now the cormorants here, they're flying cormorants. The cormorants in Galapagos are not. So that's the question of questions on how did they get there? So the origin of new species, another name for that is speciation. Speciation, which is where a new species arises from an older species. And it's really the focal point of evolutionary theory because the appearance of new species is the source of biological diversity today. So the number of species there are on Earth today is estimated to be, who knows? I think the described, like actual discovered and described species may be about, what is it, 1.8 million? But it's estimated that there could be, 10 to 100 million or something like that species. A lot of them have not been discovered. Yeah, so, well, in order to claim evolution, you have to explain how new species originate in addition to how they evolve. So we already know how they evolve: mutation, natural selection, genetic drift, gene flow that we discussed last time. But how do new species originate? So we call that macroevolution, change above the species level. And there's two basic patterns that you can distinguish. They're known as anagenesis and cladogenesis. Cladogenesis. Anagenesis is as such. You have a population of birds. I don't know what they are. They kind of look a little bit like lovebirds, but I'm not sure. They're probably just a generic bird. Anyway, so this is a population of a species, And eventually, over generations, it goes through so many changes that over time, probably thousands, perhaps millions of years, it develops into a new species. How do you determine species? That's a good question. We're going to broach that in a moment. Another way is that, oh, here's this population, but it probably split somehow. Maybe there was a volcano or an earthquake, or one group just simply beetled off somewhere and migrated for whatever reason. So these are offshoot populations. Now, this one, it didn't change. It remained the same morphologically, but this one became quite different. Look at this tail. It's a beautiful tail. Considered a different species over time. And you only get diversity with cladogenesis. With anagenesis, you just get one species which changes, but it's still only one species. With cladogenesis, now we have two species. So we have increased diversity. Increased diversity. And this is an accumulation of changes. Mutations. And the reshuffling of genes, generally through sexual reproduction. So then what are we going to define as species? What shall we define as species? Well, there's a few concepts. One is the biological species concept. It's probably the most commonly used. Species is a Latin word. It means kind or appearance. And the biological species concept is such that a species is a population or group of populations whose members have the potential to interbreed, emphasis on interbreed, in nature and produce viable, emphasis on viable, fertile offspring. Yeah, but they can't produce viable, fertile offspring with members of other populations. So you're separating this particular species by with whom they mate. I'm pretty sure it's with whom. The other day, somebody corrected my English, so I said to them, "Okay, knock knock," and they said, "Who's there?" And I said, "Two," and they said, "To who?" And I said, "No, that's to whom." Yeah, don't correct my grammar. Anyway, there is similarity between different species, sometimes quite marked similarity. So these, for example, are meadowlarks. They're both meadowlarks. They're both in the same genus, Sternella, but they're two different species. They're distinct biological species because their songs and other behaviors are different enough to prevent interbreeding. So they look the same, but their behavior is quite different. There may be quite a bit of diversity within a species even. Yeah, humans, for example, we're very similar in appearance, but we have differences, particularly external differences like skin color and hair color. They are very superficial differences caused by a very slight difference in genes. Nevertheless, there can be quite striking morphological differences between species, as we saw, or between individuals of the same species, as we saw with the Cuban snails. Quite different. So reproductive isolation is the existence of biological factors that impede members of two species from producing offspring. And there are a combination of reproductive barriers at different stages. One is the pre-zygotic stage, and one is postzygotic, so prezygotic, before the zygote is formed. A zygote is a fused egg and sperm. I could have just said a fertilized egg. That's a zygote. So a prezygotic barrier impedes or stops mating between species or hinders the fertilization of ova if they attempt to mate. Postzygotic prevent the hybrid zygote from developing into an adult. So there's pre and postzygotic barriers to reproduction. So here are a few. Habitat isolation. That could be a barrier. So in other words, these two snakes, one is a dry land individual and one exists in the water almost exclusively. So they have different habitats and don't come across each other to mate. In this case, we have temporal isolation. So they're active during different times of day, or they may be in reproductive state in different times of the year. So seasonally, they may have different timing for reproduction. So these are two different badgers here, a nocturnal badger and a diurnal badger, diurnal being active during the day. Another type of isolation that prevents species from mating with each other is behavioral. These are blue-footed boobies. And they're on the Galápagos Islands. They might be elsewhere too, but I'm not sure where. And their behavior is to lift up their feet to show their potential mate how beautiful and vibrant their feet are, which attracts the other individual immensely. And they think, whoa, those are really nice feet. And they literally walk around, and I can't really show you, but they lift their feet one after the other. Look at my feet, look at my feet. So that is a mating attempt, but mating can occur between some individuals, like these flowers, for example. The pollen from one could quite easily fall on the stigma of another plant, but the other plant does not have the right binding site for that pollen. So it will not bind there. Gametic isolation is such that the sperm might get to the egg, but the egg has these kinds of binding receptors, proteins, to which the sperm would bind before fusion. And the sperm has these kind, and they just won't bind to the egg. Yeah, so this is the egg, sperm. Let's see. I'll call them incompatible proteins for binding. Well, if a zygote is formed, it could be quite inviable. I think it's inviable. Could be quite inviable. So it might not even develop at all. Or the hybrid itself may not be fertile. So for example, if you breed, which you can, a horse and a donkey, you get a mule. But a mule can't breed with other mules. And I don't believe a mule can breed with either a donkey or a horse. And there's a polyploidy reason for that. Or possibly the hybrids break down. They just The hybrids themselves don't survive to reproduction. But, and you may have seen this flaw already in the biological species concept. You're thinking, well, Maria, what about asexual organisms? They can't fall under the biological species concept because they don't sexually reproduce. What about fossils? Sometimes you might be able to determine, but sometimes not. Some animals, plants, you really don't know much about their reproduction. So how are you going to assign them to a species? Well, there's a few ways. These are other definitions of species, which are quite valid. The morphological species concept. So characterizing a species in terms of body shape, size, other structural features like teeth, canines versus flat, carnassials carnassials are pointy too, flat molars. So that's another concept. The paleontological species concept focused on morphologically discrete species known only from the fossil record. The ecological species concept is such that it views a species in terms of its ecological niche, which is also very valid because you pretty much know you're not going to get new species if there aren't new ecological niches or new ecological opportunities. And then the phylogenetic species concept defines a species as a set of organisms with a unique genetic history. So an organism and its relatives, its ancestors, I should say, its ancestors. Yeah, so those are some other concepts of species or how to define species. So how can speciation occur? Well, it can occur with or without geographic separation. Allopatric means living apart. So here are, sorry, let's go back. This is the original population. Some kind of drying out episode separated this initial species of fish, original species. The populations got separated, and each subpopulation now has a different gene pool. So each separated population has a different gene pool. And that gene pool is the substrate on which evolution will occur. I mean, you can only change genes that are already present. The substrate. Oh no, I forgot what I said now. The substrate on which speciation occurs. So that's allopatric. So the two populations are separated geographically, but sometimes they're not. They may just be in the same lake. Speciation occurs anyway, but it only occurs if they end up being reproductively isolated. For whatever reason. So this is a good example of the three-spine stickleback, whereby one stickleback population got to be really small. So the small fishes chose the other smaller fishes for reproduction, and the larger ones chose the larger ones. And eventually there's quite a dichotomy of size in that population. So allopatric means other country. Yeah, gene flow between two separated populations is interrupted or reduced. So two or more geographically isolated subpopulations. So here's an example of chipmunks. A harrisii lives on one side of the canyon. A leucurus lives on the other side of the canyon. They are separated by the canyon, the original population. So how do these gene pools diverge? I think I'll write that on the previous one because there's room here. So gene pools diverge. Oopsie. Give me strength. By mutation, all the things we talked about. Sexual selection, a type of natural selection. Genetic drift. So those mechanisms will work on these isolated populations. With their own gene pool. Can you witness this? When you consider, oh my goodness, it takes generations upon generations for enough change to occur to consider one species having changed into two species. It seems like it would take a very long time, but you can use organisms that have a very short generation time. So these are fruit flies, and we've studied fruit flies in our class, which is cool. So this fruit fly population, this is the initial population, and they were separated. They were separated to exist on a starch diet, and these ones on a maltose diet. Yeah, and so after many generations, so in this case, it wouldn't take that long for many generations, maybe 3 or 4 months at the most, natural selection resulted in divergent evolution. In other words, populations raised on starch digested starch more efficiently. So they just began to digest starch more efficiently. Whatever enzymes there were to do that were selected for because those individuals survived. Those raised on maltose digested maltose more efficiently. So then the researcher, Dodd, put the flies from the same population, so starch and starch, or from different populations, so starch and maltose, into mating cages and measured mating frequencies and found this. Males and females together. So this is an example of the number of matings between starch and starch individuals. This is the number of matings between maltose and maltose individuals. And these are the maltose starch individuals. So that is from mating frequencies in the experimental group. In the control group, they were in the same population. So if they were the same population, 18, different populations, 15, different between the same, 15 and 12. So not that much difference. And flies taken from populations adapted to the same medium were as likely to mate with each other as flies from their own populations. So regardless of whether So these flies are more likely to mate with populations that were bred in the same medium. Yeah, it's not absolute. No, it's not absolute. There was some mating between starch flies and maltose flies. So you can witness this kind of divergence in speciation occur, although you wouldn't call them new species just yet. Sympatric means same country, geographically overlapping. And that does happen. Yeah. But what occurs is some kind of reproductive barrier. Between the sympatric populations. So an example in BC is the three-spine stickleback. So that's a good example. So there was the marine species. Although interestingly, they have been known to speciate allopatrically as well as sympatrically. And I think I did talk to you about this before. The continent was covered in ice. So British Columbia covered in ice. The ice melted. So I'll tell the story here and I'll write it down. BC covered in ice 15,000 years ago. Covered in glacier. Ice it's really heavy. You know, it's like how thick? A kilometer, more than a kilometer thick. Very heavy. So pushes the continent down. So after the ice receded, isostatic rebound, I think, isostatic rebound, lifting the coast and isolating seawater in lakes. So only those individuals that could withstand fresh water would survive. And the stickleback is one of those kinds of fish. So you've got a lake. And you've initially got your marine species. What's happened over time, interestingly, is now there are these large sticklebacks that hang around in the pelagic zone, and these really small sticklebacks that hang around in the benthic zone, and they don't interbreed anymore. So are they considered new species yet? I'm not really sure. And the last time I checked, they were considered subspecies and not individual species, but they're on their way, perhaps, to becoming new species. So sympatric speciation. So one aspect of speciation is attributed to reproduction and accidents during cell division. So polyploidy is the presence of extra sets of chromosomes in cells that's just due to accident during cell division. So what happens is, here is a cell with, I'm just going to put 2 homologous chromosomes here. And when it divided, well, sure, these 2 during meiosis ended up in different cells, but oh, these ones did not. So sometimes this is called non-disjunction. You get more than the normal set of chromosomes. So this is a good example, goat's beard. It happens quite often in plants. Goat's beard. So this one is 4N, so 4 sets of chromosomes. These ones are 2N. This one is 4N, a different set of chromosomes, and 2N. So these are different species of the same genus. Some of their difference is just attributable to the number of chromosomes that they have, because at some point there was non-disjunction. So an autopolyploid is an individual that has 2 chromosome sets or more than 2 from a single species. Failure of cell division. So here's a diploid plant. 2N is 6. 4N is 12. But the gametes produced, because there was a non-disjunction, the gametes are diploid. Chromosome duplication occurred. This individual is a 4N, but the gametes are 2N, which is very interesting. So these are called tetraploids when there are 4 sets of homologous chromosomes. I don't know, we haven't talked about this for a while. So just to review, N equals haploid. So if we're talking about humans, our gametes, eggs and sperm, each have 23 chromosomes for humans. All the rest of our cells besides our gametes are 2N. That's a diploid. And for us, that's 46 chromosomes for humans. And 4N is having 4 sets of homologous chromosomes. That's an autopolyploid, same species. The potato flower, for example, is an autotetraploid. So this kind of non-disjunction can be induced, and that has resulted in many agricultural plants. So non-disjunction may be induced in agriculture. Yeah. Wheat is an example as well, wheat and rye. Those are examples of autotetraploids that were induced. And it turns out that they're very hardy. So for whatever reason, as a tetraploid, they're a very hardy species. They can withstand cold temperatures, for example. An octoploid hybrid is when this hybrid is a culmination of 2 tetraploids. So the gametes were tetraploid. They came together and they formed an octoploid. Strawberries are octoploids. Trout, sometimes polyploid can occur naturally, but it can also be induced. So what they do is they heat shock fertilized eggs. So what happens is you cross a diploid and a tetraploid, it produces a triploid, and they are sterile. And you use that kind of method if you didn't want the individuals to have offspring, because maybe you're trying to prevent them from breeding with wild stock, for example. Yeah. An allopolyploid is a species with multiple sets of chromosomes derived from different species. So in this case, we're getting hybrids. Species A, diploid. Diploid number is 4. But there was a meiotic error, non-disjunction, so it wasn't reduced. Here's a gamete, still has the 4 chromosomes. Here's species B, 2N is 6, but produced a normal gamete. they fused. When they fused, you've got a hybrid with a very unusual number of chromosomes, 7. Well, this hybrid managed to produce some gametes itself. It probably couldn't fuse with its own individuals, the other hybrids, but it might have fused with a normal gamete from the other species, and then you have a viable fertile hybrid again. Possibly that's a little bit more rare, but it does happen. So for example, oat, it's induced, or not induced, but selected. Oats are allohexaploids. Wheat is also an allohexaploid. Yeah, so sympatric speciation can result in the appearance of new ecological niches or new ecological opportunities, which is the case with the three-spined stickleback and also the case of cichlids. So I hope that someday you get a chance to see Ian Stewart's Rise of the Continents. He talks quite eloquently about cichlid fishes. So Lake Victoria, which is a huge lake in Africa, but it has dried and undried several times, and it's resulted in a crazy amount of speciation. So this is a bit of a convoluted kind of thing, but it shows that these fish they mate with species that are subjected to specific types of light. So quite similar to the fruit flies that were subjected to either maltose or starch. Yeah, very interesting. This is Lake Victoria. It's quite an enormous lake. But there's a lot of studies in Lake Victoria, so it's worth looking into if you're interested in speciation. So since it dried repeatedly, I think there are now 600 or more different species that all arose from one originally. Quite an interesting study. So in summary, allopatric, new species forms while geographically isolated from the parents. Sympatric, there's an emergence of a reproductive barrier which isolates a subset from the parent species. So what if you get a geographic isolation on a fairly big scale? That will be known as adaptive radiation. It's the evolution of diversely adapted species from a common ancestor upon introduction to new ecological, I'm going to call it ecological, no, it says environmental, it's the same thing. New ecological opportunities. Yeah, so in this case, there is a lot of radiation. So in the Galápagos Islands, we've already talked about a little bit. If you wanted to look into the Galápagos more, actually, tortoises are very interesting. And they've come up before in our conversations. So you find tortoises who could very easily have gotten to the Galápagos on, well, I guess they would have gotten there on rafts because they're land animals. Yeah, and then they ended up on different islands. And the tortoises on these different islands are different. The different islands, different tortoises. Hawaii is a really good one to study too. So the Galapagos and Hawaii have actually some things in common, although Hawaii is much, much further away from land. The Galapagos Islands are 1,000 kilometers away from land, so not that far as bird flight goes, or organisms arriving there by raft over the ocean or drifting by wind. But the Hawaii is a little more isolated. Nevertheless, both are about 1.5 million years old. So the Galapagos is also an archipelago. And Hawaii is an archipelago. So how Hawaii formed and the Galapagos is that there is what's known as a hot spot from the ocean floor, volcanic hotspot, volcanic action. And the volcanic action forms an island, crust pushed up through the water, and then the plate moves. So when the plate moves, the island ends up over here, but over the hotspot, a new island starts to form. That's how an archipelago is formed, over hotspots. And Hawaii is one of those. Yeah, so instead of tortoises, this is a showcase of a certain kind of plant, a debauchia. And on different islands, you find different species. Why? Because populations were cut off from their parent populations and they encountered new ecological opportunities. So two things happen. One, populations cut off from parent populations. For the sticklebacks, the population was a marine stickleback population. And new ecological opportunities. I mean, if something is already occupying a niche, it's very unlikely that a new individual with these great adaptations will just move right in and take over. That's probably not going to happen. So yeah, there's the Galapagos Islands. Here's Ecuador and the Galapagos. It's 1,000 kilometers in this direction. It's also about 1,000 kilometers from Costa Rica in the north. So a couple of sources of seeds and birds and various kinds of animals that might have arrived by raft. Yeah, here are the various volcanoes that were formed from the hotspots. Very, very interesting place. I hope that you get an opportunity to go there at some time in your life. There is a lot of colonization there, so a lot of people live there, but the population is very careful to keep some things pristine. So on this island, there really isn't any habitation. But that doesn't prevent poaching. So there's actually poaching of sea cucumbers there, unfortunately. Okay, genomics. So we are now able to identify specific genes that are involved in speciation. So genomics has not only allowed us to do that, but also to identify and distinguish between species. And how fast does it happen? Well, in a fossil record, there's lots of episodes in which new species seem to appear quite suddenly in the stratum. And then they persist unchanged through several strata, and then they disappear. And that term, Eldridge and Gould called that punctuated equilibrium. So apparent stasis punctuated by sudden change. Is that reality? Well, possibly in some cases when there's a catastrophic event like volcanoes or sudden climate change like we're doing now, but sometimes it might just be an absence of the fossils or even the strata forming in between those times. So this would be a punctuated equilibrium model. You see these butterflies for a while, and then all of a sudden you see this kind of butterfly. This one persists, but there's a sudden change at this time, and now you have different butterflies, one which persists. Gradualism model just postulates that there will be gradual change, that these isolated populations will go through some allele frequency changes from one generation to the next, and they will accumulate very gradually. Yeah, most change is not, most change, like here we're looking at morphological change, but most change you can't even, is not even fossilized. Like you can't, for example, fossilize behavior. So if behavior changes, well, there's no fossil record of that. You can't detect chemical changes, biogeochemical changes. So these are things that are not fossilized. So macroevolutionary changes accumulate through many speciation events. It's the cumulative change during thousands of small speciation episodes. This is like macroevolution. And most structures evolve from previously existing structures, even though it may seem as if that were not possible. So the eye, for example, you look at the eye, the lens and cornea eye that humans have and that squid have, and you're like, well, it functions so nicely, but how could it have evolved? Well, did we ever talk about the evolution of the eye before? We didn't. I did in my other class for sure, but
the documentary showed it. You didn't talk about it specifically, but the documentary did show how it evolved.
Did. Good. Okay. So you've been exposed before. That's good. That's nice. So we can see increasingly complex structures that have a similar function. So they are adaptive at all these various stages. So here's just a patch of pigmented cells. Here's an involution of pigmented cells that can show the direction, for example, of predators. Here's a cavity that's filled with fluid and a little hole in the membrane, a pinhole eye. So the nautilus has that kind of eye, which exists today. The eye with a primitive lens, I think that's a marine snail that has that. So you can see various kinds of eyes in nature. There's not just one kind of eye. Some of them are simpler, like the pigmented eye. So this is called an eye cup of a mollusk. But the complex camera type eye, this is a squid eye. It's very similar to a mammal eye in that it has a lens, it has cornea, and it has a pupil that can expand. These two different lens and cornea eyes, they evolved down two separate lineages. It's very interesting. So evolved in invertebrate lineages and invertebrates. So how do species become different species from a common ancestor? There are genes that program development. So this is another way that change can occur. Something that happens during development. The rate of development, the timing of development of certain structures, and the spatial pattern of changes in the adult form. So it's best to see what that looks like. Heterochrony is a change in the rate or timing of developmental events. Let's see what that looks like. So this is allometric growth in humans. Wait, where's my heterochrony? Okay, well let's look at humans. Different growth rates in humans. It's called allometric growth, proportioning of body parts. So a newborn baby, for example, its head is a quarter of the size of its body, and when development occurs, eventually it becomes about 1/10 the size of the body. So the proportions have changed in development. These different patterns of development can lead to very, very different looking adult features. In this case, we have a chimpanzee fetus and a human fetus. And they do look a little bit different, for sure. But it's not until they become adults that you really see the difference. For example, in the jaw. So there has been a different pattern of growth of the timing of different parts of the skull. So back to heterochrony. Let's look at salamander feet. Salamander feet, here's a ground-dwelling salamander. There are its feet, longer digits. The tree-dwelling salamander, shorter digits, but extensive webbing. And webbing is something that you and I even have as embryos. We have webbing between our fingers, and then programmed cell death gets rid of the webbing. That's something that occurs during development. But in the case of these salamanders, there is more or less programmed cell death of webbing between digits during development. They say that phylogeny recapitulates embryology. Very interesting. In some cases, with reproductive development, the sexually mature species retains body features that were juvenile structures in an ancestral species. So this is called paedomorphosis. So instead of losing the gills and becoming, that looks like two big eyes, instead of losing the gills and becoming a terrestrial salamander, this one retained its gills. And that's quite common. Yeah, so genes can also alter the placement and organization of body parts. And that is by a set of genes known as homeotic genes. They determine such basic features as where a pair of wings or where a pair of legs will develop on a bird, or how a flower's parts are arranged. So it's a spatial arrangement. And one product or one class of those homeotic genes are called Hox genes. And that provides positional information on the development of fins in fish and limbs in tetrapods. So here's a tetrapod. Here's a chicken leg bud. The expression of that Hox gene occurs at that particular place in the body of the developing chicken embryo. And here's a zebrafish. That's the bud of the fin. Different positions. So vertebrates from invertebrates, you can associate with alterations in Hox genes. We're not going to get into this too deeply. It's a bit complex, which is okay, but in this case, the Hox genes have actually been mapped. So most invertebrates have a cluster, the Hox complex. They're the colored bands here. Yeah, and they direct the body parts of invertebrates. But apparently, about 520 million years ago, there was a duplication of the Hox complex. So that might be associated with the origin of the first vertebrates, perhaps. Yeah, so a duplication. And then there was another duplication, a second duplication. So now there's 4 clusters, and 4 clusters are found in modern day vertebrates. They occurred probably 425 or so million years ago. Yeah, it could have been a polyploidy event that caused that to happen. So that allows, because now you've got not just one set, but 2 sets of these Hox genes, and it allows the development of greater structural complexity, like jaws, for example, and limbs. Yeah, so the vertebrate Hox complex contains duplicates of lots of the same genes in invertebrates. And one thing to remember, it's very important that evolution is not goal-oriented. Sometimes, if you look at the fossil record, it kind of seems like it is. So for example, we're looking at horses. This is an ancestral horse. It's quite small, actually. And eventually, through evolution over a long period of time, let's see, this is the Eocene, so that's 55.8 million years ago, to the present, so more recent horses. It looks like they became larger and larger. Well, the only reason they became larger and larger was because large just happened to be an adaptive thing. But it's not always. Sometimes small is more adaptive, easier to hide. So there's no goal in evolution. Whatever is selected because it happens to be there and it suits the environment. Yeah, so trends result when species with certain characteristics endure longer and speciate more often, but that doesn't imply that there's an intrinsic drive. There's no intrinsic drive toward a particularly successful phenotype. And that's where I'd like to stop, and then we'll have a break.